In this video lecture, I will be talking on granulocytes. Granulocytes originate from multipotent hematopoietic stem cell in the bone marrow. And as can be seen here, this is the multipotent hematopoietic stem cell. It differentiates into self-renewal cell and differentiation cell. The differentiation cell further differentiate into lymphoid progenitor and myeloid progenitor, where granulocytes come from. I will start by giving just short accounts on neutrophils. Neutrophils are granulocytes, and they are the abundant around 40 to 70 of circulating white cells. They have 5 days lifespan, and as can be seen here, 3-looped nucleus. Neutrophils are the first cells to migrate to site of infection, and they are attracted to site of infection by damage associated molecular pattern and pathogen associated molecular pattern. Neutrophils kill pathogens by 3 ways. First, phagocytosis. It is either complement mediated or antibody mediated. And as can be seen here, this is a neutrophil. They have complement receptor number 1 and 3 service molecules, and also they express Fc gamma receptors. So the complement molecules on the surface, they mediate complement mediated phagocytosis, whereas the Fc gamma receptors, they mediate antibody mediated phagocytosis. Also, neutrophils, they can kill pathogen by expression of granules, and as can be seen here, these are the granules from the neutrophils. So neutrophils express platelet activating factor, reactive oxygen species, proteases, elastase, myeloperoxidase, cathelicidin, defensin, and matrix metalloproteinase. So these, they mediate killing of pathogens. Also, neutrophils, they have the capacity to kill pathogens by what is so-called netosis. Netosis, it means that neutrophils modify their chromatin and they produce filaments or fibers outside the cell. They look like a trap. They are trapped because they are very sticky and have the capacity to trap pathogens. So they are associated also with histone, DNA, myeloperoxidase, and proteinase 3. However, in some instances, these traps or these filaments or fibers, they trap platelets, and accordingly, they will lead to thrombosis. For example, in case of COVID-19, nets or neutrophil or extracellular neutrophil traps, as can be seen here, they occlude pulmonary vessels by blood clots and lead to a complication in COVID-19. In lupus nephritis, nets are the source of autoantigen activation and also activate the complement and will lead to formation of blood clots within the glomeruli. Nets in liver, as can be seen here, it will lead to damaging of hepatocytes and lead to chronic inflammation. In case of rheumatoid arthritis, also neutrophils, as can be seen here, this is the joint, maybe a small joint, and it is strictly forbidden for cells to come and settle here. But however, in case of rheumatoid, as it is an autoimmune, these cells will come and settle here, and they die by apoptosis. And one of the physiological process during apoptosis is formation of sertraline. So this sertraline will be trapped by the extracellular traps of the neutrophil, and this will lead to exaggeration or of the condition, and will lead to more complication and more inflammation. As the sertraline here, it will act as an antigen. Also, in case of autoimmune ulcerative keratitis, autoimmune ulcerative, this is usually stromal cell, which is long-standing ulcer, and it will lead to formation of long-standing ulcer and ulcers which do not respond to treatment. So we suspect that it could be stromal cell, and it is due to netosis. So now I am going now to talk about eosinophils. Eosinophils are granulocytes. They are around 1 to 6% of the total white blood cells. So the nucleus has two loops, as can be seen here. This is one loop and this is another, and with reddish or pinkish stained granules with eosine, as can be seen here. And so they target, usually eosinophils, they target helminths or parasitic worm. Eosinophils, they circulate for 8 hours. So they will be available in the circulation for 8 hours. And following that, they stay in the tissues for around 8 to 12 days. So they have very long lifespan in the tissues. As I mentioned that eosinophils, they have an anti-helminthic effect, and as can be seen here, these are eosinophils. They express receptors known as Fc epsilon R1 receptors, and these are specific for IgE antibodies. So these IgE antibodies are raised against some molecules on the surface of the worm. So the IgE antibodies will bind to the worm, and it will bind to the Fc epsilon R1 receptor. This binding or this interaction will lead to production of major basic protein by the eosinophils, and also eosinophils, they produce eosinophil cationic protein and eosinophil derivative neurotoxin, which is toxic to the worm. So by this way, they can kill the worms. So eosinophils also, it is similar to neutrophils. They have capacity to produce extracellular traps, as can be seen here. These are sticky filaments from the modified chromatin, and also it has capacity to trap different pathogens. Basophils are granulocytes. They are less than 1% of total white BCs, around 0.5, so they are the least common of the total or of the white blood cells. As can be seen here, this is a nucleus. It is horseshoe nucleus with dark purple granules, and basophils are loaded with histamines. And as can be seen here, they produce proteases, and these proteases from basophils, they recruit monocytes, neutrophils, and eosinophil to the area of infection. And also they produce heparin, which prevent blood clotting, and produce histamine, which promote inflammation. And also they express on the surface Fc epsilon R1 receptors, which are specific for IgE antibodies. So once the allergen bind, this will release of the mediators. And also they produce cytokines, for example interleukin number 13 and interleukin number 4, and these they have a capacity to induce class switch to IgE antibodies. So as I mentioned that interleukin number 4, as can be seen here, interleukin number 4 from activated neutrophils induce M2 macrophage. This is M2 macrophages arginase 1 that kill helminths by depletion of arginine. Also, as I mentioned that the secreted proteases recruit monocytes, neutrophils, and eosinophils to the area of infection. And these proteases also, they have a killing effect on round worms or nematodes. Also, basophils, they have the capacity to produce extracellular traps known as bits from their modified chromatin. So basophil, as can be seen here, they express cytokines, histamine, heparin, proteases, and they express IL-4, which induce M2 macrophages arginase 1 that kill helminths by depletion of arginine. And basophils, they have an anti-helminthic effect against the nematodes, and also they have the capacity to produce BETs or basophil extracellular tracts. Basophil extracellular traps. Thank you very much, Professor Ahmed Bola.